Hello everyone, this is Kunika Chandra from Shivaji College, Delhi University. Currently, I'm in the second year of my course BSc Physical Sciences with Chemistry, and today, as a part of the teacher internship program, I will be covering a topic of organic chemistry called amino acids in this lecture. So before we start, let's have an overview. So the lecture is divided into two parts. The first part would be covered in this video. We will be discussing about amino acids and their basic classification. The second part would be about synthesis, where we'll be doing it by two methods. The third is the reactions, also called the chemical properties of the amino acids. So the first thing that comes into our mind is what are amino acids, like how they are present inside our bodies. So for this, we will be taking an example of human hair. If we take a single strand of hair, we'll see that it is actually made up of a protein called keratin. So this single structure of protein is actually made up of small building blocks called amino acids, and these are the amino acids that we will be covering in this lecture. So this is the basic structure of an amino acid. It contains two functional groups, the first being the amino group NH2 and the second is the COOH, that is the carboxylic group. It also has a side chain and the carbon is called the alpha carbon. As we saw in the previous slide, if we hydrolyze protein, we will see that it is made up of small building blocks called alpha amino acids. So now when we know what are amino acids, let's see how they are classified. In nature, there are 20 amino acids which are present in almost every protein. So these 20 amino acids are divided into two parts. The first being essential amino acids. Essential because these are some amino acids that cannot be made inside our bodies, so it becomes very necessary to take them in the form of diet. The second is non-essential amino acids. They can easily be prepared in our bodies. That's why they're termed as non-essential. Moving on to one of the most important topics of amino acids is how they are prepared. The first method that we will be using is the Gabriel thalamide synthesis. This is one of the methods to remember is to prepare primary amines as well. So looking at this reaction, which is the basic reaction, we see that we have to add thalamide into an alpha haloester, hydrolyze them to get the alpha amino acid. So let's get into the mechanism of it. The first step is to introduce the thalamide group in alpha haloester. So how do we do it? First of all, we take potassium thalamide and an alpha bromoester. Since potassium thalamide has a K positive and alpha bromoester has a Br minus, they would get both attached and separate out as KBr. And so we see that the thalamide group gets attached to the alpha haloester. This is our step 1. The second step is the basic hydrolysis, in which we will be using a base in the reagent called NaOH. So what happens is this NaOH will cleave this bond, and this would get separate out to make alcohol. In step 3, we have acidic hydrolysis, where the product that was formed in basic hydrolysis is treated with an acidic reagent called HCl, and this will propel the formation of phthalic acid and alpha amino acid by breaking this bond. So this is how we get the amino acid. Let's see how we can prepare alpha amino acid through a second method, which is the Strecker synthesis. Seeing the basic reaction, we see that if we add ammonia and HCN into an aldehyde, first of all, we'll get alpha aminonitrile, and when we process it further, we'll see that we can prepare alpha amino acid. So, getting down to the mechanism, the first step is the condensation of aldehyde and ammonia to form imine. So we are taking an aldehyde and an ammonia, as we know that ammonia has lone pairs in it. So what happens is, first of all, the electronegative oxygen will take the electron pair to itself, leaving the carbon electropositive. Now, the ammonia, having the lone pair, would attack this electropositive carbon and make this intermediate. Now from this intermediate, it will rearrange itself so that we can take out water, and that is how we'll get imine. In the second step, we are adding HCN to the formed imine to form an alpha aminonitrile. So let's see how it happens. The nitrogen will take the electrons and leave the carbon electropositive. Now we know that HCN is having a positive charge and cyanide is having a negative. So the cyanide get attracted to this electropositive carbon and we'll get cyanide in this manner. In the next step, we'll see that the negative charge on the nitrogen is being attracted by the hydrogen, and that is how NH2 gets formed. In the third step, we'll hydrolyze the alpha aminonitrile to produce alpha amino acid. So as we know that if we hydrolyze cyanide, carboxylic acid gets produced. So this is the same that is happening in this reaction as well. And now I have written amino acid in its Zwitter ionic form, which we will be doing in the further slide. So when we have studied about how amino acids are prepared, let's see what type of reactions do they show. So in the previous slides, as we saw, that amino acids is actually having two functional groups, the first being amines, that is NH2, and the second is COOH, that is the carboxylic group. And because of the two functional groups, amino acid is showing a variety of reactions. The first is the reaction due to the carboxylic group, that is the esterification. Now, esterification means a reaction we do to produce esters. So looking at the basic reaction, first of all, we're taking an alpha amino acid, and as we do in an esterification reaction, we will be adding alcohol and a catalyst. The first thing that will be produced is ester salt. Now, if we want to produce a free ester from it, we will add either aqueous Na2CO3, moist AgOH, or Ag2O. So let's see the reaction. We are taking glycine, which is an amino acid, and ethanol, which is an alcohol, and we have got the ester salt over here. Now to make the free ester, we are adding AgOH, and in the reaction AgCl and water would get expelled, and we will get ethyl alpha amino acetate, which is an ester. So let's see what the amino group of an amino acid show in its reaction. So the reaction it shows is acetylation. Acetylation is the addition of the CH3CO, also called the acetyl group, into the alpha amino acid. So according to the basic reaction, we see that if we add an acid chloride or anhydride into alpha amino acid, we'll get the acylated product. First example I'm taking with the help of acid chloride. If we add it into glycine, which is an alpha amino acid, we'll get the N-acetylglycine after the expulsion of HCl. This second example I've taken with the help of acid anhydride, and in this case, instead of HCl, we'll get acetic acid in the byproduct, and the product N-acetylglycine would be formed, which is same, and we can see that how the same product can be obtained using two different reagents. And this is how the acetyl group, which is the CH3COO group, gets attached in the amino acid. Third type of reaction that amino acids show is a special property of them, in which they can actually react with metal ions to form insoluble complexes called salt. So in this reaction, if we see that if we add amino acid and a metal ion like copper oxide and heat it, we'll get an insoluble complex called the salt. And amino acid can actually react with a lot of heavy metals like Ca2+, Ag2+, to give such insoluble complexes. In this reaction, I've taken glycine in copper oxide, and so we will be getting a deep blue colored salt, which is also called as the copper salt of glycine. So that ends the first part of our lecture on amino acids. Let's take a quick recap at what we covered. The first we did was about amino acids, the basics of it, like how they're present inside our bodies and the basic classification into essential and non-essential. Next, we moved on to the synthesis that we did by two methods, Gabriel thalamide and Strecker synthesis. Next, we did some reactions that the amino acids show, also called the chemical properties. First we did was esterification, which is shown by the carboxylic group of the amino acid. Second was acetylation, shown by the NH2 group. And the third is complexation with metal ions, which is a unique property of all alpha amino acids. So I hope this lecture was clear, and thank you everyone for joining.